So thank you all for being there, either physically or remotely. I will present my PhD work that is entitled From the Mechanistic Modeling of Cancer to the Interpretation of Models, Clinical Applications, and Statistical Evaluation. And it has been done under the supervision of Laurence Calzone, Emmanuel Barriot, and Aurélien Latouche. But first, without being exhaustive, I would like to highlight some key features of cancer. The simplest definition of cancer is an uncontrolled proliferation of cells. And you have multiple mechanisms that are required to activate this uncontrolled proliferation. There are only few biological examples there. So angiogenesis, you should also enable replicative immortality. So there are many biological hallmarks there. There are multiple protections you have to blow out if you want to trigger this uncontrolled proliferation. So regarding cancer, there is one first layer of complexity because you have different mechanisms of proliferation that you have to consider all together in order to have a proper understanding of cancer. But there are other layers of complexity. You have different organs. You may have a brain tumor, a lung tumor, for instance, so it means a different biological context. And even for a given tumor, you may have tumor heterogeneity. For a given lung tumor, for instance, one side of the tumor or the other side of the tumor may experience a different behavior. And it explains many clinical behaviors also. On top of that, you also have, of course, different patients, and you have many important inter-individual variability. You have many differences between one breast cancer patient and another breast cancer patient. So that's a very important layer of diversity. And last, we also have different data types. It's not a feature of the cancer itself. It's more related to the techniques we have to look at the cancer. It's like pairs of glasses you use to look at all those different complex layers. But we will look mainly at DNA, RNA, and proteins, and we will see that we can interpret them differently. How to handle this complexity? Usually, in many scientific domains, we try to use some models, and I will try to use the kind of models I will talk about. And cancer models, as other models usually, represent the system in a simplified way. So you have physical models, the models you can touch, basically. So you can take some cancer cell lines and put them in a mouse or in a petri dish to grow those tumor cell lines. And it's much easier because you can perform some experiments. You can test many perturbations, many things. So that's also why it's a model. It's a convenient way to handle a complex system. On the other way, we also have formal models. In this case, we represent the cancer with mathematical and symbolic terms. But it's more or less the same mindset. You still try to gather many data, such as gene A, B, C, D there, and you try to give an interpretation, try to predict things. Still, you represent the cancer, but using mathematical formulas or formalisms. And I will mainly focus on formal models, but there are different categories of formal models. The first category can be called black box models, to be very generic. So usually it means that they are statistical models without a priori biological knowledge. You take those gene A, B, C, D, and you try to find the best mathematical combination to optimize your prediction of a given behavior, your cell death, for instance. So it's a naive algorithm, if you want. And it's really focused on prediction, and you have no embedded interpretation method, usually. On the other hand, I will mainly focus on what we call mechanistic models. That's still based on mathematical formalism, but this time we try to model the internal biological structure of the system. In this case, we can say that gene A is interacting with gene C, gene B with gene D, and gene C and D together are triggering cell death. This may be a biological knowledge we can have a priori on the phenomenon, and that's something we try to integrate in our mathematical representation of the cancer. That's a mechanistic model. And usually that's focused on understanding, not necessarily on prediction. And it allows an easier extrapolation. With mechanistic model, we can try more easily, at least, to add new components, such as a drug, for instance, that would inhibit gene A in that case. And you can try to add those components and to see what happens. Now we will focus on those mechanistic models of cancer to ask the main question of this thesis is, are mechanistic models of cancer clinically useful? I will present that in 3 parts. First, I will explain how to generate patient-specific mechanistic models, and I will explain why it is important to have those patient-specific models. Then we will use those models to answer a clinical question about drug responses. And finally, we will try to evaluate statistically the clinical benefits related to those mechanistic models. Along the presentation, I will use one common example about differences between melanoma and colorectal cancers. Those 2 cancers share some common molecular features, and especially they are very often mutated for one given gene called BRAF. So melanoma is very frequently mutated with BRAF, and colorectal cancer is sometimes mutated with BRAF. It's less frequent. So they share this common pattern. But if you try to treat them with a treatment targeting BRAF gene, such as vemurafenib, that is a BRAF inhibitor, then you observe that melanoma has a very good average response, even if there is some heterogeneity, and colorectal cancer a bad response, or at least a worse response. And that's something we would like to explain, and we will try to take that example and explain those differential responses to BRAF inhibition with mechanistic model of cancers. But first, how to generate patient-specific mechanistic models of cancer. And this method has been described in the following publication. But before talking about patient-specific models, I have to explain the kind of mechanistic models we will use, because there are different formalisms to model mechanistically a cell. And here we will focus on logical models. First, what about the systems? We want to model some cells, like in the picture, and in those cells, you have biological entities, let's say protein in this case, that interact with each other. Okay, they can activate protein B is activating protein A is inhibiting protein B. So you have those kind of network of interactions within the cell. And those entities can have varying activity. Sometimes they can be active or not active. It corresponds to different reality depending on the biological entity, but they can vary in their activity. That's what we would like to translate. And we will do that with the logical formalism. Logical formalism is a qualitative formalism that try to translate in a discrete way the interactions between biological entities. The 2 main features are the following. First, you have discrete variables, and especially in this case, binary variables. Protein A will be represented by a node A that is a binary variable, either 0 or 1, and the same for the other. So if you're 0, it means that the node is inactive, and if you're 1, it means that the node is active. And then those interactions between nodes will be translated as logical rules, so rule expressed with logical operators, and or not. In this case, for node B, we define that node B will be equal to 1 if A is equal to 0, A inactive, and C is equal to 1, C active. That's how we translate the model. Now that we have defined this model in general, we have to understand how to simulate this model, how to study dynamic behaviors. So on the left, we still have the same toy model. And now we will see how to simulate. Let's imagine we have this initial condition S0. We have A that is equal to 0, B is equal to 1, and C is equal to 0. Then you can notice that 2 different changes are possible. A can be updated because A is supposed to be 1 if B is equal to 1. In this case, B is equal to 1, so A should be activated. Similarly, you can notice that B should be deactivated, because B is supposed to be active only if A is equal to 0, it's fine, and C is equal to 1, it's not fine. So B should be deactivated. And instead of performing both updates at the same time, simultaneously, we'll perform asynchronous transition. We have these 2 different state updates, and we will have to choose the transition that will occur. To do that, we define some transition rates. There are associated transition rates to each node in our logical formalism framework. It is not the only one, of course. For the node A, you have an activating rate, okay, the rate for A going from 0 to 1, and a deactivating rate, and the same for the other node. Then, a transition rate may be associated to each possible transition. The top transition here corresponds to A being activated from 0 to 1, so there is the corresponding rate. And the same for the bottom transition. Now, based on that, we can define probabilities of transition. Here, the probability for the top transition to occur first is a ratio of the associated transition rate, the activating rate for A over the sum of the transition rate of all possible transitions. Okay, and the same for the bottom transition. So the sum of the probabilities is equal to 1. And now we can define stochastically, randomly, which transition will occur based on those probabilities. Of course, if one transition rate is higher, the corresponding probability will be higher. So this transition will be more likely or will happen quickly if you want. But now we can take a broader picture. If we focus on the first row there, you can notice that we have one initial condition, and stochastically, we chose one transition, and we move to the next step. We can do that one more time, of course, and go to the next step, and so on and so forth. Then you have a whole stochastic trajectories until a given final state that can be asymptotic, but it has to be checked, of course. Then you can compute other trajectories, because of course you can have different initial conditions, and even for a given initial condition, you may have different stochastic trajectories. So in order to explore the whole space of possibilities of the model, it makes sense to compute thousands of stochastic trajectories, and that's what we do. And at the end, we can compute the average of all those stochastic trajectories, and it can be understood as an average stochastic trajectory across the model. That's what we will use later on, and it's implemented in the software called MABOS. But now we have to go back to our biological question, because up to now I just presented a toy model with just 3 nodes. But this is the real model we will use. We are interested in BRAF, okay, the gene that is in the middle, and we want to know, of course, what are the other interacting entities. So here there are around 30 nodes. So this model has been built from literature, from knowledge in literature, in collaboration with Lorenzo Pantolini. And based on what is present in the literature, you can define this model. You can find in the literature that the RAS is activating BRAF is activating MEC, and so on and so forth. So you can gather all this information and translate that in the logical formalism. Here, the gray nodes represent the environmental conditions, the boundaries of the model. And the blue node is also especially important because it's the output of the model. It's a phenotypic readout. It's a way for us to summarize the phenotype of the model regarding proliferation. Okay, we gather information about proliferation pathways in order to understand whether the model is proliferative or not. Since we're studying cancer, proliferation is, of course, of primary importance. But this is one network. And the question we can ask is, okay, but how to explore inter-individual variability with one common network? Because I mentioned at the beginning that even for this given knowledge, we observe in real life that some patients have a good response to the treatment and other patients have a bad response. So we have to understand how could we explain those differences. To do that, we will integrate patient data into this generic structure. But since it's not necessarily easy to integrate continuous data or real data in those logical formalisms, so we'll have to process the data in order to interpret the data and later to integrate data into the models. I will focus here on 3 different data types: DNA, RNA, and proteins. For DNA, it's a bit easier because DNA information is usually a discrete information. You have a discrete mutation information. You know if the patient has something different for a given gene compared to a reference genome. Then what we do is we perform functional inference. It means that we infer the functional effect of the mutation using dedicated databases or softwares. So for instance, if we know that this specific mutation completely disrupts the protein structure, it's probably a loss of function. The protein is no longer able to fulfill its function. So it's a loss of function. We will use a 0 in our logical model, not active, no longer active. Conversely, if that's a gain of function or constitutive activation, we will translate that as a 1. In many cases, we are not able to interpret the effect of the mutations, and we are not able to assign. We just keep it blank. On the other hand, it's a bit more difficult with continuous data. And usually, RNA and protein data are continuous. We have a continuous distribution of RNA levels or protein levels for a given gene across the cohort. In this case, what we decided to do is to keep this continuous nature, but we normalize data in the sense that we try to preserve the distribution pattern, but we transfer these data between 0 and 1. And you will see later on why this interval is especially convenient for us. At the end, what does it mean? If the value is close to 0, it means that for a given gene, for a given patient, this patient is has the gene less expressed compared to the other patients in the cohort. And conversely, if you're close to 1. Now we can integrate this data into the logical models. For discrete data types, one more time, it's a bit easier. If you say, for instance, that A has a loss of function mutation, you can decide that your node A will be forced to stay at 0 for the rest of the simulation. That's what we do. And in this case, some transitions are no longer possible. So it's a very stringent way to influence the way the model is simulated. And conversely, if you say that B is constitutively active, then B will stay active during the whole simulation, and you prevent some transition to occur. It's a bit more difficult for continuous data. Let's focus on gene A and this patient N with the red profile. Here we have the value of normalized value for gene A across the cohort. So we have score between 0 and 1 for this gene for each patient. My patient N has a high value for this gene, is more expressed than the other patients. Okay, so we have something like 0.9 over 1, let's say. And now we can integrate this data in two different ways. First, we can define the initial probability of activation for A. Since we compute thousands of stochastic trajectories, we can define the initial probability for node A to start as 1. So 0.9 is, so we will have 90% of the trajectories in this case that will be initiated with A is equal to 1 for this patient. Then we can also use the data later on playing with transition rate. Since we say that based on our interpretation, A should be quite active or more active than in the other patients, we decide that we will increase the corresponding rate. We will increase the activating rate for A. Like this, we will enforce this transition and we will favor the activation of A, forcing the model to be close to our interpretation of patient data. So on the top, we have this discrete personalization that is quite stringent, and on the bottom, we have this continuous personalization that has a softer influence. The key thing to remember there is that we do not perform any model training or parameter fitting. We really interpret the biological meaning of the data, and then we integrate these data into the model. But we do not fit the model based on a given objective function or based on data. So this is the method, more or less. But now we would like to see, okay, is it possible to apply this method to a clinical problem? Because that's our topic of interest. And in this case, we will try to apply those personalized mechanistic models, to the problem of differential drug response. And it has been described in the following preprint. In this part, I will use mainly cell lines data, because we have much more data, of course, for cell lines compared to patients. I will have around 60 cell lines, 30 colorectal cancer and 30 melanomas, some of them with BRAF mutation, some others without BRAF mutation. For each cell line, we have a molecular profile, mutations plus RNA. So it means that we will be able to derive a corresponding personalized mechanistic model per cell line, one mechanistic model for each cell line. And then we also have some experimental data regarding the sensitivities of these cell lines to the BRAF inhibition, based either on a drug that can inhibit the BRAF gene or based on CRISPR screening, which is a slightly different technique that targets directly the gene to erase, if you want, the gene. But both of them are inhibiting BRAF. Then we will compare what the model predicts and what we observe in the experimental data. Schematically, here is what we do. On the x-axis, we will use only personalized mechanistic model. We compute what happens for a given cell line when we inhibit BRAF in the personalized mechanistic model, and we observe the corresponding proliferation score. We also compute what is the proliferation score for the given cell line without BRAF inhibition, and we can compute this ratio. Of course, the lower is the ratio, the more sensitive is supposed to be the cell line, according to our personalized mechanistic models. And on the other hand, we have experimental sensitivity. So that's based on biological assays that measure this sensitivity. So in this case, sensitivity means you are more sensitive if the drug really impairs your proliferation. And the metric here is built more or less in the same way. It's called AUC. If you're close to 1, it means the cell line is sensitive. And if you're close to 0, sorry, you're sensitive. And close to 1, you're not sensitive to this drug. So we'll be able to compare this and see whether it is correlated or not. And here are the correlation figures on the left. So we can see, for instance, that if we personalize the models using only mutations, we have already a decent correlation figures around 0.63. And if we look at the corresponding scatter plot on the right, you see that we have real clusters. Okay, the correlation is meaningful, okay, but still, when you look at the precise picture, you only have clusters. And here, you are just able to identify the cell lines that have been BRAF mutated at or this big vertical cluster. It makes sense to have those clusters because based only on mutations, many cell lines are similar or identical. So of course, the model is not able to distinguish. Then, if we use only RNA, we don't manage to recover the experimental sensitivity because missing mutations, we miss too much, basically, of the biology, according to this method, apparently. But if we combine mutations used as discrete personalization plus RNA used as continuous personalization, because you can do both together, then we recover a decent correlation with a much more dispersed picture, basically. Because now cell lines are different. Based on RNA, they cannot have the same profile. So the picture is much more dispersed. Here it's a bit difficult to decipher, so I will focus more on the next experiment with CRISPR. We have less cell line, and I will explain more precisely what we observe. Here for CRISPR, for the matter of definition, the metric is opposite. So we do not no longer expect a positive correlation, but a negative one. It's related to the definition of the sensitivity metric. Sorry. So using only mutation there, we have one more time a decent correlation, but still we have those clusters because the model is just able to identify some combination of mutations. Now let's look at the mutations plus RNA. And to have a closer look, so the score is better, of course, but we need to have a closer look mechanistically. So I will keep that, but I will focus on some cell lines. I will focus especially on this one, SKML24, that is around close to my mouse there, and HT144 that is there. Why those two? Those two are two melanoma that are BRAF mutated. So in terms of molecular profile, they're very similar. On the left, I put an excerpt of the molecular profile. So first and third row, you can see those two cell lines. And in terms of mutations, indeed, they are just mutated for BRAF. They have this classic constitutive activation of BRAF. So since they have the same molecular profile based on mutation, for the model on x-axis, we expect a similar sensitivity. But experimentally, that's not the case at all. We have a very different sensitivities. If we add RNA, we observe that SKML24 is pushed to the left, which makes sense because we expect indeed SKML24 to be more sensitive. And HT144 is pushed to the right towards less sensitive cell lines. On the top left here, you have the more sensitive cell lines. What does it mean in terms of mechanisms? If we look at RNA profiles of the cell lines, we notice one key difference. HT144 has a very high level for CRAF RNA, and SKML24 has a very low level for CRAF. What does it mean? If we look at this schematic representation of the model, here is what happened. Here we have BRAF. If you inhibit BRAF there, you decrease, of course, the BRAF activity. Then you decrease MEK activity, you decrease ERK activity, since all those nodes are activation, so positive links. So you decrease ERK, and then here you have a negative feedback loop. So if you decrease ERK, you increase EGFR, you increase RAS, and then you can circumvent this BRAF inhibition through CRAF, through this pathway. And in this case, for HT144, since CRAF level is high, the model is able to perform this activation of CRAF. You can activate CRAF pathway due to this feedback loop, so you can circumvent the BRAF inhibition, and therefore you're not sensitive because you can circumvent the inhibition. And the opposite, for SKML24, you cannot circumvent because you cannot activate CRAF, so you're very sensitive to BRAF inhibition. And that's what we observe. And we think that one of the main interests of those mechanistic models is to be able to really decipher cell line per cell line the potential mechanisms that explain the sensitivity. So there are other examples in the manuscript, but we can discuss several mechanisms like this, and at least we can try, based on the model plus the profile, to understand what happens. But all in all, an important point is that apparently adding RNA on top of mutations it makes sense, and it allows us to add another layer of another relevant layer to the model. So to conclude on this part, it's possible apparently to generate qualitative and personalized mechanistic models without fitting parameters by just interpreting the data and integrating the data. Of course, there are some limitations and questions. It's data saving because since you don't train the model, you don't need many data basically, and it may be interesting when we compare with some machine learning approaches. It's in some cases with very few data, but it's very intensive in terms of expert knowledge. It's difficult to build those models, and we can wonder whether it is easily scalable or not in theory and in practice to multiple targets simultaneously. We can also wonder what are the right proxies for node activity. Here I use mutation data and RNA data, but you can also wonder, is it relevant to use protein data? It was not available in this data set, but in general, we can wonder what is the best data to infer the node activity and how to integrate that in a relevant way. And last but not least, all these models clinically useful, because I showed there that we can interpret those models for a clinical problem, at least in the preclinical data, but still a clinical problem in terms of the drug response. But is it useful? And that's what I would like to answer in this last part about statistical evaluation of the clinical benefits that has been described in the following preprint. What I would like first to highlight the links between those parts, the previous part and this one. If you have a cancer model that is mechanistic or not, by the way, that is capable of predicting the response to several treatments, then consequently this model is able to predict the best treatment. In this case, if you have two patients with different molecular profiles, you perform a mechanistic model, personalized, to see whether the orange drug, the red drug, or the yellow drug is the most efficient. And then you can say, okay, based on the model, apparently for this model, the yellow drug is the best drug, or the red drug is the best one. So if your mechanistic model is able to predict the response to several treatments, then it is a precision medicine tool. Precision medicine in the sense that for each patient's molecular profile is associated a treatment recommendation made either by a physician or by a treatment assignment algorithm. A treatment assignment algorithm is a very generic word, but it can be a mechanistic model. It can be a cancer model in general. And on the bottom, you have an example of a treatment assignment algorithm in a precision medicine trial, Shiva, that has been performed in real a few years ago. So treatments associated to alterations. Later on, those precision medicine algorithms will just be defined as PM, precision medicine, a function that associates a treatment to a molecular profile. Now, how to quantify the clinical benefits of these algorithms. But before explaining how to quantify the benefits of a complex precision medicine strategy, I would like first to remind how to evaluate the effect of a very simple treatment. Then we will refine this method, but we first have to understand how to do that in the simplest case. If you want to assess the effect of a treatment A on an outcome Y, in spite of a confounder C, what would you do? And what does it mean? If I take a very simple example, if your treatment is sparkling water, and you want to see if sparkling water is better than plain water in terms of lung cancer. Okay, I don't think that sparkling water is triggering lung cancer, but I don't know. If I want to test that, I can. Okay, I have a look at people drinking sparkling water, drinking plain water, and I compare. But the issue is that if we have a confounder, imagine that for whatever reason, smokers drink more sparkling water. I don't know why. Then if that's the case, since smokers have more frequent lung cancer, you will have a confounder because the confounder, smoking status, will impact both the treatment and the outcome. So you will not be able to understand easily what is the causal link, the precise influence of treatment on the outcome. That's the main problem, one of the main problems when you want to evaluate the effect of a treatment. In real life, to deal with that, we can use randomized clinical trials. So you take eligible patients and you randomize them. So randomly you put them either in the treatment arm, A is equal to 1, or in the control arm, A is equal to 0. And then you can compare the average outcome, the average Y, when A is equal to 1, and the average outcome when A is equal to 0. You can compare those two because thanks to the randomization, the proportion of the confounder is balanced in both groups. So that's why randomization is useful there. But what happens if you want to assess this causality, but with observational data. Observational data means that that's just the data that have been generated, but not been randomized, or at least not been randomized according to your wishes. In this case, in my observed population, let's imagine I have 3 untreated patients in red and 3 treated patients in green. I have those patients, I have this data, but unfortunately, the smoking status is unbalanced. I have more smokers in the untreated. So how can I deal with that? It's too late to randomize if you want. So either I just compute the association, I compute the average outcome for the untreated patient, the average outcome for the treated patient, but in that case, it's just association. It's not causation because we still have this confounder issue. So we will try to move from association to causation, and to do that, we can have a kind of ideal perspective about that. What you would like to do in an ideal world is to compare the whole cohort left untreated, all patients red, versus the whole cohort treated, all patients green. In this case, of course, the confounder will be balanced in both groups. Of course, it's theoretical because you don't know what would have happened to this patient, for instance, when untreated, because you don't have access to this value. It's called a counterfactual outcome. You would like to know what happened to this patient if, contrary to the fact, it had been left untreated. So in order to infer causation, we will need to think about those counterfactual outcomes. And those counterfactual outcomes will be noted like this. It's y as a function of a. And in this case, this counterfactual outcome is y if a is equal to 0, even if that's not the case in the real data. But of course, it's not a magical trick. To do that, we will need some assumptions and some models. There are different causal inference methods to try to infer those counterfactual values. You can use an outcome model, for instance, y as a function of a and c. In this case, you can feed this model and use this model to infer the counterfactual outcome. Okay, you feed the model based on the observed data and use this model to infer what would have happened on the other patients. In this case, this method is called standardization or G computation. It's based on an outcome model, a model of the outcome. You can also use a treatment model. So you model the treatment A in function of the confounder C. In this case, this method, at least the one we'll use, will be called IPW for inverse probability weighting. Or you can use both models to have a more robust method, even a method called doubly robust. And the one we use there is called TMLE, for targeted maximum likelihood estimation. But let's just remember that we have 3 different methods with slight technical differences, but still all of them are based on the same statistical assumptions that are called exchangeability, consistency, and positivity. They are statistical assumptions that need to be validated in order for the causal inference to be valid. Now, with those methods, we will be able to simulate some trials. We can do as if we perform randomized clinical trials. But what are the trials we would like to do in the case of precision medicine? If we have our precision medicine algorithm there, first we focus on eligible patients, only the patients that have a treatment recommendation based on the algorithm. And then we randomize them either in the precision medicine directed arm, the top one, in which each patient is treated according to his or her molecular characteristics. The green patient is treated with the green drug, and so on. And then we'll compare this precision medicine directed arm with different kind of controls. The first possible control that we imagine is just a reference treatment. Okay, all patients are treated with, let's say, the same standard chemotherapy. In the precision medicine arm, usually you use more targeted treatments that target a specific gene. But here, this reference treatment is just a broad spectrum chemotherapy, let's say. So if you compare precision medicine versus reference treatment, you have a first causal contrast, the first causal effect you can measure. We will call this one CE1. Another possibility is to compare precision medicine with physician's treatment, because in your observational data, of course, patients have been treated based on the physician rationale. They have not been treated randomly. So we can compare your precision medicine arm with what has been done by the physician. It will be called CE2. And last, you can also compare your precision medicine arm with a random assignment of the same targeted treatments. So you use the same treatments, the same targeted treatments as in the precision medicine directed arm, but you assign them randomly. So in this case, you really assess the value of the assignment algorithm itself. It may be a bit more difficult to imagine on an ethical point of view, but it exists in some current clinical trials. Now we can try to simulate those clinical trials. But to do that, we will need to refine a little bit the previous framework I presented, because we still have C, the confounder, A, the treatment, and Y, the outcome, but we will need to add the version of treatment, K. C will be, in our case, the genomic covariates, okay? The covariates, the biomarkers C that are used to define your precision medicine treatment. A is the treatment. In this case, A is equal to 0 correspond to the generic treatment, the chemotherapy. And A is equal to 1 mean one of those precision medicine drugs, one of those targeted drugs. But then it's not enough to consider them in the same pool, because of course, that's the essence of precision medicine. You have to consider the drug itself, the precise molecule. So we introduce the version of treatment, K, and if you are in the precision medicine arm, of course, K will be determined by your covariates C, your profile, your molecular profile, and then we have the outcome Y, that is, for instance, cancer relapse, and we will consider that continuous or binary. Now we have a new counterfactual variable. Y is no longer a function of A only, but also a function of K. And for each patient I, we have a precision medicine treatment, KPM, that is a function PM of the genomic covariates C. Now we can derive the estimators of those causal effects, the statistical estimators. If we focus there on CE1, there's a comparison between precision medicine and reference drug. So imagine that for the reference drug, we have one unique version of treatment, K0. We can compute, we can simulate the precision medicine arm on the second row there. So here we have the average outcome Y. When A is equal to 1, okay, we have a precision medicine drug. And the version of treatment is not random, it's KPM. It's the precision medicine version of treatment that is defined basically by the function PM. And then you can compute the standardized estimator, that is a sum over all possible covariates, a weighted sum over all the possible covariates. And here in the middle, for this Y as depending on A, K, and C, this will be inferred based on the outcome model, because it's standardized estimators, one more time, or based on the model of the outcome Y. Similarly, you can derive statistical estimators for the reference treatment, the last row. It's even easier, of course, because in this case, you have only one single version of treatment, so that's much easier. In a similar way, you can also derive statistical estimators for all other causal effects I mentioned previously. Here is just one other example with CE3, so the random assignment of targeted treatment. In this case, we can define the distribution of treatments, G, that is completely random with equal probabilities in that case. And we can define, one more time, the average value of Y, the outcome, when your version of treatment follows the distribution G, that is, in this case, a random distribution of the versions of treatment, and that's the same kind of formula. So this is a statistical framework, but now we would like to test whether it works or not. And we first test that on simulated data. So first, we simulate data with all counterfactuals. So we simulate thousands of patients, and for each patient, we have some confounders, let's say mutation status, biomarkers. Then we define the observed treatment, the one assigned by the physician, and the personalized treatment that are based on this precision medicine algorithm. Then we define the outcomes, okay, the counterfactual outcomes. And we define the linear models, for instance, that underlie those counterfactual responses, and we can derive the observed response. And then for the simulation, for the real simulation, we sample a subcohort, or we sample many subcohorts, and for each subcohort, we can do two different estimates. In green, we use all counterfactuals to derive the ground truth, but this is not possible in real life because we need to know all the things about the patients, even impossible things in real life. And using observed data only, we use our causal inference methods. Here, a very simple and very brief example of simulation scenario. In this case, we define a scenario with two different genes, C1 and C2. each improving the response to one of two drugs, Ka1 or Ka2. And here it's just the distribution of observed treatment. So treatment assigned by the physician, but in this case, do not depend on the genomic covariate. That's a simpler case. Then we define some models, in this case, just linear models that underlie the counterfactual responses. So we define what happened to our patients. In this case, we define that C1 is improving the response to K1 and C2 is improving the response to K2. Okay? In this case, the lower the response, the better it is. It's an analog to tumor volume. And we can have a look at the performance. Here for CE1, the comparison between precision medicine and reference treatment. On the left, in blue, you have the true effects, so the effect based on counterfactuals, the ground truth. On the right, in red, you have the naive method, so using only observed data, but without any correction for the confounder. And in the middle, you have the causal inference method, the three I mentioned previously, TMLE, standardization, STD, and IPW, inverse probability weighting. So if we focus on the mean absolute error between the different methods and the true estimates, we can notice that the highest bias, the highest gap between true values and estimated values is, of course, for the naive method that do not correct for the confounder. And then we observe an improvement with causal inference methods, and especially with the TMLE method that is supposed to be more robust. So we are closer to the ground truth using those causal inference methods. Since we have many possible simulation scenarios, we also designed an interactive application with a very short overview there in order for the user to be able to define what is the number of genes, the number of treatments, the kind of response you want to simulate, the population size, and all the parameters of the simulation. And there is basically an infinite number of simulation scenarios. So that's why we provided an application to test that. And it's quite important for us in order to be able to understand how the method behave in different situations. And last, we try to apply that no longer on simulated data, but on real data, and especially here on preclinical data. We found a very interesting data set, in our opinion, that is called patient-derived xenograft. What does it mean? You have a tumor, a patient tumor, and then you cut this tumor in several pieces, and you implant each piece in different cloned mice. Then each mouse is treated with a different treatment, and therefore you have different mice with the same tumor, or at least pieces from the same tumor, but all treated with different treatments. So it's a very good proxy to counterfactuals. It's a way to access something you will never access otherwise with real data, which is counterfactuals. You know the response for a given tumor to different treatments, such that we can use this real data exactly in the same way as we used the simulated data, because we have access to all the counterfactual responses. So we designed a very simple mock algorithm, precision medicine algorithm to test. We decided that all patients with KRAS, BRAF mutations will be treated with binimetinib. All PI3K mutated patients will be treated with a corresponding inhibitor. And let's keep in mind that this is a very simple example, but this kind of algorithm can correspond to a mechanistic model or to a cancer model in general. In this case, the example is much simpler, but conceptually, it's exactly the same thing. Then we have exactly the same kind of results, or at least the same pattern of results, but we can observe that the improvement in performance is modest. There is slightly improved performance with our causal inference methods. And it's important to mention there that here, in order to observe this slightly improved performance, we no longer use linear models as previously, because previously all these outcome model, treatment model that we use in our causal inference methods were linear models. In this case, if we use linear models, the results with causal inference method are very similar to naive method. We are not able to correct the effect of the confounder. So here we needed to use some machine learning nonlinear methods, and especially in this case, random forest, because otherwise we are not able to correct for the effect of the confounder. And one more time, TMLE, the doubly robust method, appears to be the most efficient. To conclude on this part, we have statistical methods adapted to precision medicine in order to evaluate those precision medicine algorithms using observational data. There are one more time some limitation of questions, of course. First, we have to be very careful with the misspecification of model. It had been illustrated just in the previous slide with this linear model versus random forest issue. We have also potential positivity issues depending on observational data. I mentioned positivity as one of the assumptions required to perform causal inference, and this one is maybe especially problematic if in your observational data the assignment of treatment have been deterministic. And last, we can also wonder, as a perspective, if it's possible to extend this kind of work with data from human clinical trial. So all in all, what I presented there is one path to go from mechanistic models to clinical evaluation. But it's only one path, because I just summarized there some of the choices of some of the markers of this work, because there are many other possibilities. We decided to use qualitative logical models. We make the hypothesis that we can biologically interpret the data before integrating them. We use cell lines to validate those approaches, and we focused really on mechanistic explanation of the mechanisms, of the differential rate responses. Sorry. And last, we decided to evaluate those models, seeing them as precision medicine tools. You can imagine other perspectives, but that's what we have chosen there. And we decided to simulate clinical trials with observational data. Those are the different markers, I think, of what I presented. And of course, there are many other paths that are possible to answer very similar questions. And I just mentioned those open questions to conclude. I presented there what we can call single layer models, because we focus on models of molecular networks. But you can imagine multi-scale models that connect to other scales, to the whole tumor, to pharmacokinetics, or to other kinds of models on other scales of modeling. I also presented qualitative mechanistic models, but there are also plenty of quantitative mechanistic models, and it's also a whole field of research, of course, and with many interests. And I also focused there on the evaluation of models from the point of view of clinical benefits, mimicking the clinical trials, but there are also other possibilities to focus more on algorithmic performance and prediction performance, and that's a different point of view about that. And I will stop there. I just would like to acknowledge one more time my supervisors that are there, of course, Laurence, Emmanuel, and Aurélien, the members of the jury, of course, also the lab members, either physically or remotely, and all the peoples and relatives that came, and it's really a pleasure. Thank you.